is graded correctly.
Sure.
Either using this key or the one on the pilot. The student came in this morning and she thinks it might be authorized on it.
Okay. Oh, okay. Are the slides that you wanted on there?
Yeah, it's just checking. Looks good. I'm ready. How about you guys? The one bit of just general information. If you need to take the makeup exam for the class, the chance to do that is Tuesday of next week between 11:30 and 2. It'll be in 104 of the biology building. If you haven't missed the first or the second exam, don't worry about it. If you did miss it for some reason, that's your one chance. Okay? And then I am aware that there's a holiday coming up next week, right? And that a lot of you are going to be leaving and so forth. And so people have asked me a few times now, are we really going to have a class on Tuesday morning, the week of Thanksgiving? Don't you know what's going on? And here are my thoughts. I'm going to be out of town on Monday. I have to testify in a trial in New York City. And I'm expecting to have no trouble getting back, right? But it is a busy travel time and sometimes there are issues. If I have a problem with that, I'll get word out to you by midnight on Monday that I'm not going to be around. But I'm thinking I'll be here, all right? So don't worry too much about that. And I do plan on being here on Tuesday, but I'd like to remind you that these lectures are available on Pilot. There's a integrity recording of each of these lectures. And if you don't want to be here on Tuesday, I don't mind talking to an empty room and letting you guys watch this as you're digesting your feast on Thursday afternoon. All right, so don't feel like you have to be here. If you have somewhere else to be, I appreciate that. Just catch up with what I will have talked about to the empty room. Don't pay any attention to the cricket noises in the background, and you'll be able to get everything that you would have gotten if you had been here. And then speaking of getting things without being here, there was a little trouble with the recording of the audio on Tuesday's lecture of this week. It kicks in at about the 25 or 30 minute to go mark, right? And so what I'd like to do this morning is, just for the record, redo the beginning more or less of the lecture on Tuesday, just hit the highlights, and then finish off talking with you today about the central dogma. I'm hoping that at the end of class today, and by the way, the syllabus says that I was supposed to talk about the central dogma on Tuesday and Thursday today, so we'll be right on schedule. I'm hoping I can even get a little bit ahead of schedule so that for Tuesday's lecture, I can just do the fun part, which is the regulation of gene expression, how prokaryotes at least decide when to turn genes on and when to turn genes off. That is fun stuff, and I get a lot of kicks out of talking about it. There's some stuff at the beginning of that lecture just about bacterial genetics, mapping genes in bacteria. I hope I'll be able to talk with you about that today so that I can just get straight to talking about gene expression on Tuesday. All right, but so again, let me just hit the highlights for you of the lecture from Tuesday now that we've got the sound working again for the whole lecture. You'll remember I talked with you, I made some jokes, I said some disparaging things about Watson and a little bit about Crick, talking about how molecular biology is a dogma, something that's been handed from God that tells us very simply this: information in cells, prokaryotic or eukaryotic, information always flows from DNA to an intermediate transient molecule, RNA, and ultimately ends up being used to make proteins. All right? That's the central dogma. It's just that flow of information, DNA to RNA to protein. And Some of the earliest insights came from from Beadle and Tatum, who were doing some experiments with bread molds, and what they found is that they could get these inborn errors of metabolism for these bread molds. They'd irradiate them, expose them to radiation, and they'd look at the mutants that they had, and some of the mutants that they got weren't able to live on what's called minimal media. Minimal media is pretty much just water, some salt, and a little bit of sugar. Okay? So again, they found that they could make mutant bread molds that could not live on minimal media. They could only live if they supplemented the minimal media with a biomolecule or two. Right? And some of the ones that they started working on at first looked as if they could only survive if their minimal media was supplemented with a specific amino acid. So they were able to do some neat experiments with those mutant bread molds. Again, if they grew them in minimal media, they'd get no growth, right? They just couldn't make do with salt, sugar, and water the way that bread molds usually can. But they found some mutants that would grow if they were grown on minimal media plus a chemical called ornithine. They had others that would grow if they were fed minimal media with another organic molecule called citrulline. And they found a third class that would only grow if it had minimal media and the amino acid arginine to work with. And on top of that, they found that these guys could grow if you fed them arginine or citrulline. These guys could grow if you fed them arginine or citrulline, but not ornithine. And these guys could grow only if you gave them arginine. And the long and the short of it ends up being this. It looks like from these experiments, and it's been borne out many times since, but it looks like from these experiments that making the amino acid arginine must be a three-step process. And that the last step in the process is an enzyme that converts something to arginine. Okay? The something that gets converted to arginine must be citrulline. And another step in the process involves transforming something into citrulline. That something that gets transformed to citrulline must be ornithine. And what these Beadle and Tatum folks had done when they were doing these experiments at Stanford is they isolated mutations, mutant versions of these molds that were defective in being able to make each of those different enzymes for that three-step process. And so they were able to map out the metabolic pathway. And again, this sort of follows on the footsteps of discussions about inborn errors of metabolism. These mutations were faithfully transmitted from one generation of these bread molds to another. And it gave rise, it really sort of locked down this idea that for every gene, there's an enzyme or a protein, if you want to be a little bit more liberal about it, right? And so it really pinned down this idea of one gene, one enzyme. And that's a paradigm that we're still working with today to a large extent. I think it's fair to say that that is true. One gene, one enzyme. That's definitively true for prokaryotic organisms. When you get into the more complicated eukaryotic organisms, especially the very complicated ones like you and I, it's not strictly true anymore because I've mentioned it on Tuesday. I'm going to talk with you about it in more detail today. But we have about 35,000 genes, and it looks like we can make about 250,000 proteins because of the way that we alter the information in our genes after they've been transcribed. So for eukaryotes, especially complicated ones, it's not necessarily one gene, one enzyme, but certainly genes are what gives rise to the information that's used to make enzymes. All right, so there's Beadle and Tatum's experiments. There's a picture of the central dogma. Again, these arrows are shorthand for processes. The first process is called transcription. The second process is called translation. Transcription is catalyzed by RNA polymerase, and translation is catalyzed by ribosomes. What I did on Tuesday is I talked with you a fair bit about what's going on here with transcription, and I stopped pretty much at the point where I was about to start talking with you about translation. So I did about half of the central dogma, the front half of the central dogma. Okay? Today, I want to refresh your memory a little bit about what's going on with transcription, but then also focus a bit on the upshot of translation. So let's get into a few words about, again, how this plays out inside of a cell. In prokaryotes, it's a pretty simple process. I tried to emphasize to you on Tuesday that transcription and translation happen at the same time in the same place. So for a prokaryotic organism, that's how it's done. And I even suggested to you, let me say it to you very plainly, that as an RNA polymerase is making an RNA copy of a gene as it's transcribing, there literally are ribosomes sniffing around by the butt of the RNA polymerase looking for things that they can translate. Okay, so the ribosomes in the RNA polymerases are pretty much working at the same place at the same time. As RNA comes out of the RNA polymerase, ribosomes are there working on translating it. Same place, same time. And that's in contrast to what's happening in eukaryotes, where transcription happens exclusively in the nucleus. So if I were to ask you on an exam, the final exam, gee, where would you might expect to find active RNA polymerases? There's one definitive correct answer to that: in the nucleus, right? RNA polymerases only hang out inside of eukaryotic nuclei. If you're a eukaryote, that's where that's where your RNA polymerases are. So transcription happens only in the nucleus, but translation happens only in the cytoplasm, only after the RNAs have been exported from the nucleus out to where the ribosomes are. So for eukaryotes, transcription happens in one place at one time, and translation happens at a different place and a different time. The consequence of that is pretty simply this: we have the ability as eukaryotes, and again, complicated eukaryotes like humans take full advantage of this opportunity, but we have the ability to alter the information content of our RNA molecules before ribosomes see them. So unlike in prokaryotes, where whatever's spelled out at the DNA level is essentially exactly what the ribosomes see, in eukaryotes we can change those transcripts and change their information content. And we do that routinely in at least 2 and usually 3 different ways. And I'll tell you about those in just a minute. All right? So, pretty big difference in that sense, because transcription happens in the nucleus and translation happens in the cytoplasm. For eukaryotes, because of that, it's often useful to talk about things like a pre-mRNA. Sometimes you'll hear pre-mRNA used equivalently with HNRNA. Heterogeneous is what HN stands for. But pre-mRNA, HNRNA, primary transcript all mean the same thing. That's what comes off the DNA by the RNA polymerase. What the ribosomes see is different. They see the processed RNA or the messenger RNA, the mRNA. That's the only thing that gets out into the cytoplasm by the ribosomes. All right, so let's just talk briefly about what goes on with this general idea of transcription. Remember, DNA is a double-stranded molecule. The 2 strands are anti-parallel to each other. One strand goes 5' to 3' in one direction. The other goes 5' to 3' in the opposite direction. The 2 strands are complementary, meaning that if you tell me the nucleotides on one strand I can tell you with certainty what the nucleotide sequence on the other strand will be. Okay? What an RNA polymerase needs to do is tease apart this double-stranded DNA molecule and use the information on one of the strands to make an RNA copy of the other strand. Right? So there are two strands. One of them is going to be used as the template for the RNA polymerase. And what's going to happen then is that this is the template strand, and RNA polymerase will come along and it will synthesize the new RNA molecule in a 5' to 3' direction. It starts at the 5' end and it moves to the 3' end. Okay? It's moving along the template strand 3' to 5'. But the RNA synthesis is 5' to 3'. Everybody focuses their attention on the baby new RNA molecule, right? It's nobody pays any attention to the template strand. When you have these discussions, it's always, oh, look at the cute little baby RNA molecule coming out the butt of the RNA polymerase. Isn't it darling? Everything's about the RNA molecule, and so that's why we're always talking about RNA synthesis being 5' to 3'. Okay? I understand the RNA polymerase is actually moving 3' to 5' on the template strand, but the focus is on the new RNA molecule. And that again gets synthesized 5' to 3'. I should tell you, about 10 years ago when I was teaching this class, there were 3 guys, must have been, Air Force officer types, right? Always wearing their freshly pressed blue shirts. And at the end of the semester, one of them said to me, hey, Professor Crane, do you know how many times you said 5' to 3' during the course of this class? And I don't know, I'm guessing like 50, 60. It's like 362. And it's like, wow, you guys have a lot of extra CPU capacity in your heads if you're keeping track of that while I'm talking about other things. But again, I do say 5' to 3' a lot. And I think that might mean that it's kind of important. So if you're fuzzy or uncomfortable about it, don't just gloss it over. Sit down, look at a picture like this, wrap your head around it. I've tried to point out to you what it is at the molecular level that makes the 5' end of the molecule physically different than the 3' end. It's every bit as important as understanding the difference between left and right. Okay, so when you're trying to read a book, one of the very first things that you stumble upon is, oh, the information only makes sense if you go in one direction. That's certainly the case for molecular biology. So you make this pretty baby new RNA molecule, and then ribosomes can come along and look at stretches of 3 nucleotides in a row. Those 3 in a row we call codons, and each codon corresponds to a different amino acid. Okay, and so those can get put into place by ribosomes during the course of translation. Here is the genetic code. Let me emphasize this. It is a universal genetic code. All living things on this planet use that same code. In my mind, this is a pretty compelling argument against the idea of an independent creation for each of the living things. There's no reason that you have to have the same code. The reason we have the same code is because all living things share a common ancestor, and this code must have been the same code that common ancestor billions of years ago worked out and worked with. And we've inherited it faithfully ever since. So we've got this universal genetic code. You can see they're arranged in codons here, and you can translate conceptually using this code. If I were to ask you what amino acid does the codon CCU code for, right? Well, here's CCU. I know the answer, proline. That's going to be the answer whether I'm talking about E. coli, dogs, or people, right? Again, it's a universal genetic code. And I'll draw your attention to a couple of obvious things, I suppose, on this genetic code. Notice that of the 64 possible triplet codes, it's how many there are in this table. 3 of them are reserved for a special purpose. They're effectively punctuation marks. They tell the ribosome, stop translating here. So when the ribosome comes upon that triplet code, it says, whoa, that's the end of the line. This protein is done. We don't need to translate anymore. No more amino acids need to get tacked on. So aptly named a stop codon. They tell the ribosome, stop. Okay? And equivalently, more or less, or similarly might be a better way to describe it, there is one codon that's reserved as a start codon. It also is the codon that tells ribosomes to put in the amino acid methionine. So protein synthesis begins at start codons. Proteins, the first amino acid in any baby protein will be a methionine. And then if you need a methionine somewhere else within the length of the protein, you can trot out that codon again, but translation starts at AUG and it ends at one of the 3 different stop codons. Okay. I'm going to talk with you more about mutations after I've talked with you about translation, so we'll get back to that idea. I hope I impressed you some of the cool things you can do with this knowledge. Because the genetic code is universal, if you took the firefly gene that causes their abdomens to glow, it's an enzyme called luciferase, if you took their gene and you put it into a tobacco plant, you can get a tobacco plant that glows every bit as much as a fruit fly's abdomen glows. Tobacco ribosomes are translating and making proteins exactly the same way as fruit fly or firefly abdomens are. All right? They're the same gene using the same code. If I take that gene from one organism and I put it into another, I can get funky things like glow-in-the-dark tobacco or glow-in-the-dark pigs. All right. So, transcription. Here's the deal. I've talked with you about it at a relatively high level. I'll talk with you about it at a slightly lower level. Here's the deal. You got your double-stranded DNA, okay? We've got this blobby thing here that's the star of the show for transcription, RNA polymerase. RNA polymerase has to do some important work, and it's not just a business of making an RNA copy of the template strand of a gene. Also important for an RNA polymerase to do is to answer 2 really important questions. And this is essentially going to be what the whole lecture on Tuesday of next week is about. RNA polymerases have to answer 2 questions. Is this the start of a gene or not? Right? And if it is a gene, is this a gene that I need to transcribe or should transcribe or not? Okay? So is it a gene or not? And if it is a gene, should I be making an RNA copy of it or not? That's resting heavily on the shoulders of RNA polymerases. And a big part of how it is they solve that problem, they answer those first 2 questions, is they scan along DNA molecules looking for specific nucleotide sequences, that taken together we call a promoter. When an RNA polymerase finds a particular string of nucleotides, in prokaryotes, it's about a dozen nucleotides. When the RNA polymerase finds that specific string of about a dozen nucleotides, it sits down on them and says, this is a promoter. And that string of nucleotides says not only is it the start of a gene, but the gene's that way. Okay, so it gives some orientation information to the RNA polymerases too. And so the RNA polymerase then says, okay, this is a start of a gene, a promoter. Now maybe I should start doing some transcription. The first nucleotide that the RNA polymerase puts into a new RNA molecule, just as it's getting started, is called the +1 nucleotide. It happens to always be an adenine, an A. Right? We'll talk more about that on Tuesday. But transcription starts at the +1 nucleotide, and we can say that that transcription start point is the beginning of a transcription unit. And the RNA polymerase then will proceed going 3' to 5' on the template strand, but synthesizing RNA 5' to 3' until it gets to the end of the gene, or in other words, at the end of the transcription unit. So it makes an RNA copy. And it has to do that by denaturing or separating the two strands of the DNA, and then progressing along, again synthesizing the RNA from the 5' end toward the 3' end. Here you can see there's some complementarity, some base pairing that's going on. And at the end of the process, the RNA polymerase falls off and starts looking for another promoter, and it releases a shiny new RNA molecule that can be used by ribosomes for the purposes of translation. I didn't say anything about this word on Tuesday, but I guess I can mention it now. It'll certainly come up on Tuesday of next week. There's a flow to all this, right? You're always moving from the 5' end of the new RNA molecule down toward the 3' end. There's this directionality, a flow, and it conjures up this sort of image of a stream. And so you'll often hear people who do this kind of work talk about things that are upstream and things that are downstream. And just as in an ecology class, upstream and downstream are relative terms, right? You always have to say relative to what. So what's the nucleotides, 10 nucleotides downstream of the transcriptional start site, right? That makes sense. I can't just say what's downstream. Well, relative to what? You need to give me a reference point, like the transcriptional start site or the transcriptional stop site or the start codon. Give me some sort of reference point, but then I can, upstream essentially translates to before or left, and downstream translates to after or right. Okay, so we have this colorful language of upstream and downstream going on as well. All right, again, I apologized for the graphic nature of this slide. When it flashed up on Tuesday, I should have deleted it for today. I didn't mean to offend you all by showing you the sorts of intimate details of what's going on with an RNA polymerase. But that's what's happening up to this point in the lecture. Let's talk a little bit about RNA processing and then really hit the ground running on translation. Prokaryotes cannot process their RNAs. Prokaryotic RNA molecules get translated right out of the gate. As soon as they get made, ribosomes are translating them. You and I and other eukaryotic organisms are different. We can process our RNAs. And what we'll do is, as the RNAs are being transcribed, the modifications to them, their information content starts to get changed pretty much right away. There are 3 different kinds of modifications that we do to our RNAs. One of those, and the first thing to happen, is the application of a 5' cap. RNA polymerase in a eukaryotic cell starts transcription. An RNA molecule begins to poke out the backside of the RNA polymerase. Almost as soon as that first nucleotide of this new RNA is exposed, an enzyme, cleverly named capping enzyme, comes along and attaches what we call a 5' cap. And it does it in a funky way. It's a guanine that gets attached by this weird 5' to 5' linkage. Not 5' to 3' like we usually talk about, but this weird sort of backwards kind of opposite way of linking things together. It's unusual. It kind of stands out. It's a 5' to 5' linkage of the guanine to the first nucleotide in the RNA molecule. Okay? That first nucleotide in the RNA molecule, by the way, is always an A, and so you attach a G that's not templated at the DNA level to that A, and that's now the new 5' end of your RNA. Weird linkage, 5' to 5'. If you look at that diagram, you're seeing there are 3 phosphates. That makes it weird too. Right, so there's a 3-phosphate sort of thing that bridges the gap between the guanine and the adenine. It looks weird. A reasonably good question to ask at this point is capping, what's it good for? Right? Why do we bother with capping? What's going on with this? The only I slipped there. The only RNAs that get capped in eukaryotic cells are the ones that are going to end up being messenger RNAs. Period. They're the only ones that get kept. And you may be wondering, well, what other kind of RNAs may there be, right? You already know there are a bunch of other kinds of RNAs. There are tRNAs, we'll talk about them in a little bit. There are ribosomal RNAs, I'll talk about them in a little bit. There are small nuclear RNAs, I'll talk about them in a little bit. There are a whole bunch of different kinds of RNAs. There' only one kind of RNA that needs to be given attention by ribosomes. Those are the ones that get the cap at their 5' end. So effectively, if you ask ribosomes, what are they good for? Well, if you ask that question, and none of you have any idea what I'm talking about. It's like in a musical. Okay, so if you ask, why are they there? Why are we capping these messenger RNAs? It's to help them be recognized by ribosomes as ones that need to be translated. So the very first modification that happens is the addition of a 5' cap. I said it quickly, let me emphasize, it's not templated. The RNA polymerase didn't see a C on the template strand that it brought a G in for. It brought in a nucleotide, and then independent of whatever that previous nucleotide was, the capping enzyme puts that G on. So you get a 5' cap. Then transcription happens. Let me just draw your attention today to some features of the lay of the land. I'll talk more about them on Tuesday. But check this thing out. Again, this is a eukaryotic concept, but it could apply to prokaryotes as well. UTR is short for untranslated region. So the RNA polymerase starts transcribing, but it starts transcribing something that ribosomes aren't going to translate. Ribosomes only start translating when they encounter a start codon. And the start codon doesn't have to be right at the beginning of the 5' end. For eukaryotic genes, it can be dozens, it can be hundreds of nucleotides before you encounter that AUG start codon. And so everything that comes before that, everything that comes between the transcriptional start site and the translational start site, we give the name 5' because it's at the 5' end of the RNA, untranslated region. Transcribed, but not translated. So, transcription chugs along, ribosomes encounter the start codon, and they proceed until they get to a stop codon. In prokaryotes, the stop codon and a transcriptional stop signal are pretty much hand in hand, but in eukaryotes, what happens is the RNA polymerase keeps trundling along well past the end of the translated part of the RNA, and another enzyme called polyadenylase comes along, recognizes a polyadenylation signal, reaches 100 or 200 nucleotides downstream of that, and cuts off the RNA molecule, even if the RNA polymerase is still transcribing away. And after it gets cut off, a couple hundred adenines get tacked on. That's the second modification that we make to our RNAs. We cap, we splice, I'll talk about that in a minute, and we clean up the 3' end by putting on this long string of adenines. Again, not templated, just a long string of adenines that get added on to give us this poly A tail. And then, I already mentioned it just a minute ago, this idea of splicing. What happens is this third modification, now we're talking about a wholesale modification to the information content of an RNA molecule. Because what will happen during splicing is parts of the primary transcript literally get cut out, and the ends opposite them get joined together. The parts that end up in the messenger RNA, this is a formal definition, if it ends up in the messenger RNA, we call it an exon. If it gets cut out of the primary transcript, in other words, isn't what's in the final messenger RNA, it's an intron, right? You're either an intron or an exon. one or the other. You're either in the messenger RNA at the end or you're not. These introns, this looks to me, frankly, like a picture of a globin gene. I have some personal familiarity with globin genes. Part of my PhD work, I was sequencing them and characterizing them. But check it out. This is a eukaryotic gene. What do you think? How do I know that's a eukaryotic gene? I'll tell you how I know it's a eukaryotic gene as opposed to a prokaryotic gene, because prokaryotes don't process their messenger RNAs. If it hasn't been clear, let me shout it out to you right now. Prokaryotes do not have introns. Okay? Let's just be really clear on that. This is only a eukaryotic phenomenon. If a prokaryote had an intron, it would have a serious problem because the ribosomes would be translating the introns, and that's just not going to work. But what's happening here is this, let's say this is the alpha globin gene. What you got here is a stretch of nucleotides that can be translated to be the beginning of the alpha globin protein. And then there's this interruption, intron. It's actually short for intervening sequence. Exon is, I think it's supposed to be excised. In a sense, that's the thing that's not excised, but that's all right. What has to happen is you have to break the RNA molecule at the beginning of the intron and at the end of the intron, and you have to link together the two flanking exons to get the final messenger RNA. Introns, right? There are, boy, don't get me started on introns, right? They're an interesting puzzle. Almost every human gene has at least one intron. There are a couple of interesting exceptions of genes that don't have introns. Pretty much of the 35,000, maybe 5 don't have introns. The alpha globin gene is kind of a lightweight. It only has 2. There are human genes. The gene that's responsible for cystic fibrosis, this human cystic fibrosis transmembrane regulator protein, all right? That gene has over 50 introns. Off the top of my head, I think it has 67 introns, so a really large number of introns. And check this out, the protein that cystic fibrosis transmembrane regulator, CFTR, the protein that gene codes for is an unusually big one. It's several hundred amino acids long. The messenger RNA is a couple thousand nucleotides long, but check this out. The primary transcript, over 1 million nucleotides long. Wrap your head around that scale, right? I'm telling you, the coding sequence, the messenger RNA, the final product is a couple thousand nucleotides long. The primary transcript from which that messenger RNA gets made is on the order of 1,000 times bigger, right? Messenger RNA, a couple thousand, primary transcript over 1 million. Crazy, right? And put this further into context, E. coli's whole genome is 4 million nucleotides long. We've got a gene whose primary transcript is a quarter the size of all of E. coli's genome, and most of that, the vast majority of that, gets left on the cutting room floor because these introns get spliced out to make the final messenger RNA. How about that? So introns are a huge deal for eukaryotic organisms. I think you might be wondering, isn't that wasteful? And I think the short answer is, yeah, that wastes a lot of ATPs. But the priorities for eukaryotes in this regard are different than the priorities for prokaryotes. Prokaryotes are all about the bottom line in terms of saving ATPs. Eukaryotes are much more interested in being able to do a wide variety of things, of being very complicated organisms that give us more abilities. And we're willing to trade off efficiency for those capabilities. Because what will happen is that what's an intron in one cell type might actually be an exon in another. So there's this interesting idea that eukaryotes have, or that utilize, called alternative splicing. What's an intron in one tissue might actually get used as an exon in another tissue. And because of alternative splicing, for us, the rule, one gene, one enzyme, doesn't quite apply, because a single gene at the DNA level can be used to make a variety of different proteins depending on how splicing takes place, depending on what gets recognized as an intron and what's an exon. I mentioned in passing on Tuesday that the exons often correspond to functional domains within proteins, and I skipped over this pretty fast because it scared me as I looked at it, but I'll tell you this. Introns get removed by an enzyme called a spliceosome, okay? And spliceosomes have to do some dicey work, some pretty high quality precision work. They need to be able to precisely recognize the beginning of an intron. I mean, you can't be off by a nucleotide or two. I'll talk about that in just a bit. And they have to be able to precisely recognize the end of an intron. You can't be off by a nucleotide or two. And they have to be able to precisely join the flanking exons together to give rise to a useful or a working messenger RNA. Spliceosomes are an amalgam. It's sort of a mixture of RNA molecules. That's what the little red squiggles are here. Those are supposed to make you think of RNA molecules. Specifically, they're small nuclear RNA molecules, snRNA. Just, good for the molecular biology trivial pursuit game. You guys won't have to worry about snRNAs aside from being aware, ooh, there's another kind of RNA. They're small, they reside only in the nucleus, and they hang out in spliceosomes. So there are snRNAs that work in concert with some proteins in about a 50-50 mix, protein and RNA, to come together to make a spliceosome. And spliceosomes do this magic of recognizing the beginning of an intron and the end of an intron and linking together the flanking exons. All that gets done by spliceosomes. I can tell you this, the snRNAs differ from one tissue type to another. And that then becomes the basis for alternative splicing. Okay, so what's an intron in a brain cell doesn't necessarily have to be an intron in a liver cell, because the snRNAs are looking for different things for those splice junctions, the beginning and the end of the introns. All right, so how about that? Time to talk about protein synthesis. Tell you, the spliceosome picture scares me a little bit. This one scares me more. That's a ribosome. And that's a ribosome kind of like dissected, looking at it the way that it's working in real life. Let me draw your attention to a couple of features of this ribosome. There is a large subunit and there is a small subunit to a ribosome. This is true for prokaryotic ribosomes and eukaryotic ribosomes. Okay? So all ribosomes have this same basic architecture. Something else that's pretty much constant across all living things is ribosomes are a mix of RNA molecules and of proteins. Okay? I mentioned that already in the context of spliceosomes. The ratio for a ribosome is about 40% RNA and about 60% protein. And there are a whole bunch of proteins, literally dozens of proteins. The number differs between prokaryotes and eukaryotes, and even between some eukaryotes, and even between some tissues in eukaryotes. But there are dozens of proteins that come together with RNA molecules. There is a large subunit RNA called a ribosomal RNA, or an rRNA. And then there is a small subunit rRNA as well. But these proteins and those rRNAs come together to make ribosomes, large subunit and small. And what will happen is the messenger RNA snakes through sort of the interface between the large subunit and the small subunit. And coming out the butt of the ribosome, sneaking through the large subunit, is a polypeptide that's being made as the RNA gets translated. So you've got an RNA molecule with a 5' end and a 3' end. Transcription happens in the 5' to 3' direction. You make the RNA from the beginning to the end. You start at the 5' end, you go to the 3' end. Translation also has an orientation. Translation also proceeds 5' to 3'. The ribosome starts at the 5' end of the messenger RNA and it cruises along toward the 3' end. As it's going along, the ribosome is bringing in tRNA molecules into one of 3 different positions, and those tRNA molecules are sniffing around for the anticodons. They're sniffing around for the codons to help the ribosome figure out which amino acid to bring in next. And as those amino acids get strung together, again, you grow the polypeptide and you have a new protein. So here's a little bit less intimidating picture of a ribosome doing its thing. You'll see what's happening here. You've got your large subunit on the top, the small subunit down on the bottom. The RNA is coming through. The ribosome is moving along the RNA from 5' to 3'. And what will happen is these things, that's a pretty crude rendering of a tRNA. T is short for transfer. tRNAs are all about what's going on at one end of the tRNA molecule called the anticodon. The anticodon can base pair with the codons in the messenger RNA when there's a good match, when the anticodon of the RNA can base pair with the codon of the RNA, things sort of click into place, and the ribosome plucks the amino acid, or plucks an amino acid, off of the other end of the tRNA and adds it to the growing polypeptide. So this is how translation is playing out and how it ends up being mediated essentially by these tRNA molecules looking at the RNA. And here, I said that was sort of a crude picture of a tRNA. This is more what a tRNA might look like if you wrote it out. Check it out. It's an RNA molecule. Okay, it's short. Usually tRNAs are 50 to 70 nucleotides long. It never gets translated. Okay, that's not what tRNAs are about. They're involved in translation, but they never get translated to make any kind of protein. They've got a 5' end, they've got a 3' end. Their 5' end doesn't get capped, right? So ribosomes aren't interested in translating them. And if you go from the beginning through to the end, right, 5' to 3', it's at the 3' end that the amino acid ultimately gets attached. It's at the bottom loop here that you've got an anticodon that's going to be feeling along on the messenger RNA. And notice how the tRNA has stretches of nucleotides that are complementary to each other. So when you first hear about RNA molecules, people usually try to keep you simple and pure and just thinking of RNA as being a straight shot, single-stranded molecule. The reality is that the nucleotides that come together to make RNA molecules are just as sticky as the nucleotides that come together to make DNA molecules. And so DNA molecules like to stick to each other because of hydrogen bonding between the G's and the C's and the A's and the T's. RNA molecules like to stick to each other because of the hydrogen bonding between the G's and C's and the A's and U's, because in RNA we use uracil instead of T-thymine. Okay? And so there's actual base pairing. I can even use this fancy word. There is intramolecular base pairing between this part of this tRNA molecule and the other part, between the beginning and the end. Look, a G is hooked up with a C, a G with a C with a G, a U with a G. Not much happening there. But a U with an A with a U, an A with a U. So you get these little stretches of double-stranded patches within the RNA molecule that give tRNA molecules this very characteristic shape. All tRNA molecules have a loop. This is a stem, that's a loop, that's the anticodon loop, and they also all have two other loops that give them, again, this sort of signature structure that all tRNAs have, whether you're a prokaryote or a eukaryote, whether you're coding, whether you're bringing in phenylalanine or arginine, they all have this same basic structure. When the whole thing folds up, so this is sort of like a primary sequence is the linear order in which the nucleotides are arranged. A secondary level of structure would be this base pairing. The tertiary structure, where it all sort of contorts and folds up together into a three-dimensional structure, looks like this. And then this is a poor artist's rendering of that three-dimensional structure. Right? So that's what tRNAs look like. And again, here's that same picture. And I'm hoping that after what I've just talked with you about, you could answer a question like this if it were to come up at some point, right? There's a bunch of questions there, really. Where's the 5' end and where's the 3' end? Notice the 5' end and 3' end aren't labeled in this particular picture, but if you recall from the previous slide, the 5' end is over here, the 3' end is over there. So the 5' end is here, the 3' end is there. Where are there some hydrogen bonds? There, 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 there. Anywhere there's base pairing. Where are there unpaired regions? Oh, this is an unpaired region. That's an unpaired region. Where's the anticodon loop? Well, there's the anticodon. There's its loop. Where's the binding site for amino acids? It's going to be up here at the 3' end. All right, so just basic anatomy of a tRNA molecule. All right, so that's what's up with translation. Ribosomes, as they're translating, are adding amino acids into a growing polypeptide. That process is called elongation. You could call it translation too if you like. And let's talk about some of the practical consequences of, well, for lack of a better word, mistakes at the level of DNA. Right? There's a pejorative word that we use to talk about these differences. We call them mutations. All right, so let's talk about mutations for a little bit. Mutations get a bad rap, right? It's like if you go up to somebody in a bar and you tell them that, hey, you're a mutant, that probably means you're going to get in a fight. I don't have personal experience, but I can just imagine how this would play out. People don't like to be called mutants. Again, they get a bad rap. Mutations really are the grist mill. It's the grist from which natural selection can get some variation to say, ooh, this is good. So some mutations actually are beneficial to a population. They change an existing gene subtly in a way that makes the gene work better in a given set of circumstances than the original version had. So mutations aren't always bad, but if you've got a fancy watch and you pry open the back of the watch case and you start poking around with, better yet, you start getting a 3-year-old to poke around inside with a crayon or a scissors or, a screwdriver, what do you think? Are the changes the 3-year-old's going to be doing, introducing random changes to the structure of the workings of your watch? Is that going to improve your watch's function, or is it going to make it less of a watch than it was before the process started? We are pretty finely engineered machines, gene machines in some sense. Any random change to the information in our genes, more likely than not, is going to make the whole system work less well. But once in a while, that 3-year-old will knock off the bit of lint that was holding 2 gears together, and now suddenly your watch will work better than it did before. Right? So sometimes these mutations are helpful. More often, because of the complexity and how the intricacy with which things are working, those sorts of things will be a problem. So let's talk some more about mutations. There's this molecule called hemoglobin. It's responsible for carrying oxygen in our blood. Hemoglobin is made of actually 2 different proteins. There's alpha globin and beta globin. Hemoglobin is a tetramer. There's 2 alphas and 2 betas that come together to hold heme which in turn holds oxygen and lets us carry oxygen from our lungs to all of our peripheral tissues. Okay? That's hemoglobin's primary job. Hemoglobin, again, is made from two different kinds of proteins, alpha globin and beta globin. Both of those proteins are coded for by genes. And within the alpha globin gene, at one stretch along the length of the alpha globin gene, is a triplet codon that codes for the amino acid glutamic acid along the length of the polypeptide for the alpha globin protein. That's the normal scenario. That's what almost everybody in this room has for both their copies of the alpha globin gene. But there is a fairly common alternative version, we could even call it a mutant version of hemoglobin, where there has been a single nucleotide change at the level of DNA. I haven't made this clear to you guys before. We got a big genome, 3.2, but 3 billion nucleotides. You change just this one, you got a situation on your hands. All right? So if this one nucleotide change happens, it could either be an A that became a T or a T that became an A, right? It's hard to tell which came first here, but if you make that one change at the level of the DNA molecule, at that spot in the alpha globin gene, you'll have a different codon. The codon used to be GAA, now it's GUA, and that codon doesn't tell ribosomes to insert glutamic acid. Instead, it tells ribosomes to insert the amino acid valine. Glutamic acid is a negatively charged amino acid. Valine is a hydrophobic amino acid. There's a big difference between glutamic acid and valine, and it really has an interesting manifestation for hemoglobin. Glutamic acid here, this is an amino acid that's on the surface of the alpha globin protein, and so it causes two adjoining alpha globins to kind of repulse each other a little bit. Those negative charges are uncomfortable, so they don't get too close to each other. The valines, instead of the glutamic acid, has just the opposite effect. It makes a little hydrophobic patch, so it causes one alpha globin to be drawn toward another alpha globin, and it causes one hemoglobin molecule to sort of stick to another hemoglobin molecule, and that to stick to another one, and you get this chain of hemoglobins as opposed to loosely bopping around hemoglobins, and the chain distorts the shape of the donut-looking red blood cells, and so instead of being cute little round red donuts, sacks full of hemoglobin, they're sort of oddly shaped donuts with things poking out of them, sometimes looking like a sickle. Those things poking out between one red blood cell and another red blood cell hook onto each other, and they give rise to clots that give rise to all sorts of problems for people with sickle cell anemia. So the whole basis of this complicated disease is ultimately a single nucleotide change. And we can talk about, well, geneticists and people who study molecular evolution will have a specific name for this kind of change. They'll call this change a substitution. It's a substitution because what's happened is the amino acid that was being coded for by the gene is now a different amino acid. It's been substituted with another amino acid. Now, not all mutations result in substitutions. I didn't say it today, I said it on Tuesday, but if you go back to that table of the genetic code, you'll notice pretty quickly that most of the amino acids actually get coded for by more than one triplet codon. There are actually 2 amino acids that are coded for by 6 different triplet codons. We have a word for that. We say there is a redundancy in the genetic code, but that redundancy then gives rise to this, is you can change the nucleotide sequence of a gene in a way that has no effect whatsoever on the amino acid sequence of a protein. All right? Make sure you're comfortable with that. It's because of the redundancy of the genetic code. But it turns out there's no difference between boy, they didn't make this easy on me, did they? But apparently GGC codes for glycine just as GGU codes for glycine. So if you change this from a C to a T, no big deal. Ribosomes, when they encounter GGC, behave exactly the same as when they encounter GGU. They put a glycine in there, right? At the level of the protein, there's no difference. At the level of the DNA, there's a nucleotide difference. There's been a mutation. Those kinds of substitutions, those kinds of mutations are called silent because you don't hear about them. There's nothing much that happens with them. The ribosomes behave as if there's been no actual change, even though at the DNA level there's been a change. Here, if you change the GGC to AGC, this is a mutation, but here, this is a missense mutation because, well, here we have, it's not silent anymore because now instead of coding for glycine, we're coding for serine. So some nucleotide changes have an effect, some nucleotide changes do not have an effect on the amino acid sequence of the protein. Here's another kind of mutation that you might have. Over here, the second codon in this sequence was AAG. If you changed that A to a U, not only does it, this is a little bit different. It's not like you've changed the lysine to one of the other amino acids. What you've done is you're now invoking a stop codon. UAG is one of the three stop codons, and when the ribosome encounters the stop codon, it says stop. So it stops translation, and what you end up with is a very truncated version of the polypeptide, right? Instead of being 4 long, now it's only 1 long. Those kinds of mutations we call nonsense mutations. They introduce a stop codon, and you lose all the rest of the information. The ribosomes aren't able to translate anything further downstream from there. And then lastly, a last kind of mutation is check out what happens if you introduce an extra nucleotide somewhere along the length of a gene. Here, what they've done is between the AUG codon and the AAG codon, they've added another nucleotide, a T, which ends up being a U at the DNA level, at the RNA level, sorry. And this one introduces what's called a frame shift mutation. It happens that this frame shift mutation is also a nonsense mutation. But let me just make sure I touched on this with you guys. Frame shift mutations are pretty devastating because if it's somewhere along the length of a messenger RNA, you add in another nucleotide, consider what the implications are for everything, and I'll use the word downstream, right? Everything that comes after that is going to be, for lack of a better word, out of whack. Ribosomes go in lockstep, codon by codon by codon. And if you slip in an extra nucleotide, all the rest of the codons downstream of that are going to be out of register. If you slip in 2 extra nucleotides, you've got a frame shift mutation. If you slip in 3 extra nucleotides, maybe not too bad. You'll still keep the same reading frame. You'll introduce 1 extra amino acid, maybe a problem, probably won't be much of a problem. But if you introduce 4, frame shift. Introduce 5, frame shift. Introduce 6, maybe a problem, probably not. Right? So the fact that ribosomes are looking so intently at 3 at a time and are being so careful to stay in that register, to stay in that frame, to stay in that reading frame, insertions and deletions are often just devastating to the functionality of a protein that's coded for by a given gene. So insertions and deletions, unless you're talking about insertions or deletions in multiples of 3, are a real bummer in the sense of the consequences for the functionality of a protein. Okay? So these are different kinds of mutations. Here's a frame shift that doesn't introduce a stop codon, a nonsense mutation. Instead, it gives you extensive missense, meaning essentially every triplet codon downstream will be something different than it was before. So check out what happened. It was AUG, AAG. AUG, AAG is still there. But now what they've done is between this U and this U, another U gets added. I think that's what's happening here. Oh, one gets deleted, so they've taken this one away. And so now instead of having a codon that is UUU, we have a codon that is UUG. UUU codes for phenylalanine, UUG codes for leucine, different amino acid. And on top of that, instead of having the next codon being GGC, now the next codon is GCU. GCU codes for alanine, GGC codes for glycine. So frame shift mutations, pretty big deal. They mess up the information content of the genes downstream of where they occur. So here they're showing you that if you took out 3, life's not too bad, right? We just have a shorter protein than you would have had otherwise. If that lysine was important to the functionality of the protein, well, you could have a problem. Okay. Those last couple of slides talking about mutations may be giving you a misunderstanding about something. And it's simply this. How big is a typical gene, and how big is a typical protein? I'll start with the second question first. A typical prokaryotic protein is a couple hundred amino acids long. Okay? To be able to do something useful, to be able to bring together the right R groups, to have a chemistry that lets you catalyze a reaction, right? You need a couple hundred amino acids as a rule. Okay? Eukaryotic proteins tend to be a little bit bigger than prokaryotic proteins, maybe 300 or 400 amino acids long instead of 200 or 300 amino acids long. But you need to have a molecule that can do useful things. You need that level of information, that level of complexity. So if we work with 300 as a good sort of like framework for the length of a protein, of a typical protein, how big then is the length of a typical gene in a prokaryote? 300 amino acids must mean that the gene is at least 900 nucleotides long, right? Because for each amino acid, there's a triplet codon at the DNA level that's spelling out which amino acids to string together. So a typical prokaryotic protein, we'll say, is 300 amino acids long. The coding sequence of the gene will be on the order of 900 nucleotides long. And then you have to factor in things like the 5' untranslated region and the promoter. And you start talking about a typical prokaryotic gene being on the order of 1,000 or so nucleotides long. Right, so that just gives you, again, the sort of sense of scale. For eukaryotes, because of those stinking introns, all bets are off, right? And the length of a typical eukaryotic gene, you know, you're starting at a bare minimum talking in terms of several thousand. And again, there are instances within the human genome of genes that are over 1 million nucleotides long. Again, primarily because of those introns and the implications associated with them. All right, well, check it out. That's the end of that lecture. I flashed this up at you at the end of class on Tuesday, so you don't have to worry about getting it again today. It hasn't changed. It's the same one. And there's those things. And now, I got 5 minutes. Let me say something to you about viruses and about bacteria. And then we'll be off and running about regulation of gene expression on Tuesday next week. Well, you guys are all home warm and in bed, and I will have fought my way in to speak to a room full of crickets. So, what I really want to talk with you about is how it is that prokaryotic genes get turned on and turned off. All right? I'm not going to do that today. That's for Tuesday. But to get there, let me cover these two things really quickly. Say a few words to you about viruses. Viruses are sort of in the gray area. The first time we met, I tried to talk with you and get you guys to wrestle with this philosophical issue. How do you know that chair's not alive? Right? Oh, what's up with that? And I suggested that over Thanksgiving, I think, you conduct some experiments with your grandparents. Are they asleep on the couch, or what's going on? We got to perform some tests. Right? Viruses, I don't know. You can get into lengthy debates. I want no part of them. Right? Don't start talking with me. Oh, I think viruses are alive, and try to goad me into a fight. I don't care. I'm not sure if they're alive or not. But viruses are sort of on the edge of being alive or dead. It's hard to say for sure. But one thing we can say for sure is that viruses have a nucleic acid genome and a protein protective coat, and these viruses with their nucleic acid genome and their protein protective coat can infect living things like a plant cell and end up replicating as a result of that. Viruses come in some interesting shapes and sizes. Right? All of them have a nucleic acid that contains their genetic information. Some of them have an RNA-based nucleic acid, an RNA-based genome. HIV, for instance, is perhaps the most notorious retrovirus that people talk about these days. Retrovirus means that the genome isn't a DNA genome, but actually an RNA genome. HIV has a single-stranded RNA genome. There are other viruses that have double-stranded RNA genomes. There are viruses that have single-stranded DNA genomes, and there are viruses that have double-stranded DNA genomes. So they come in all sorts of different combinations, but they're all nucleic acids, and they all have protein coats. Some of them are really simple, these little rod-like guys. Some of them look for all the world like Sputnik, all right? Some of them take it another level. They've got the nucleic acid here, an RNA on the inside. This, to me, I think they're trying to show you what HIV looks like. And the proteins are still there, but it's also engulfed in a phospholipid bilayer. So this is getting pretty fancy. And then these are my favorites of all, the little lunar lander module viruses. So again, the structures of these viruses can be really quite remarkable to look at. But they have that basic theme: nucleic acid to store their information, their genome, and then protein to keep that safe. Again, my favorite. Oh, great. All right. And so what happens with a virus is the DNA or the nucleic acid gets introduced into the cell. It commandeers the cell's gene expression machinery, so it stops the cell from making its own proteins off of its DNA, and it instead gets its own proteins made. An important step in this process then is going to be replicating its own genome and then encapsulating it in the proteins that it's gotten made off of its own genome and releasing them so that they can go out and infect new cells. Quickly, let me tell you something about bacteria. I feel badly about this. I'm not going to be able to do it justice. I'm going to have to come back to you guys at the start on Tuesday to talk about these bacteria. But bacteria have chromosomes, right? No big surprise. We've talked about that already. There are genes on those chromosomes. Those genes can get mapped out. They have been mapped out. The earliest way that genes got mapped out was through a trick called conjugation mapping. You guys know what a conjugal visit is? Right? Conjugation mapping is you get two bacteria to start having sex, and then you interrupt them. And you look to see which genes got transferred over. The longer you let them go, the more genes can get transferred over, and so you can map genes out by conjugation mapping. This is a diagram of conjugation mapping. This is another picture of conjugation mapping, and this is a question based on conjugation mapping. You'll find this question in your lecture notes, okay? And it's derived from this idea of conjugation mapping. I'm going to ask you a conjugation mapping question on the final exam, all right? They're kind of fun, in a weird way, puzzles to sort through while you're gearing up for the football games after you've had some turkey, all right? But try to wrap your head around these. The lecture notes give you a sense of what's going on. You may actually just be able to figure it out on your own from a question like this. I'll try to talk you through it at the beginning of class on Tuesday, all right? But have a great weekend. I'll see you all in December. Some of you maybe next week.